Okay, this is the Professor, CBMD PhD, and today's Friday, August 4th, 1923. It is 4:00 and 9 seconds, a.m. Eastern Standard Time, and today's research, epigenetics. So let's come to it here. Researchers find an epigenetic key that unlocks common deadly cancers. Date July 31st, 1923. Source, Rockefeller University. In skin subaparietal epidermal stem cells turn on SOX9, kickstarting a process that ultimately activates cancer genes. As scientists have never fully understood how this doomed outcome ensues at a molecular level, but now researchers have revealed the mechanisms behind this malignant turn of events. As SOX9, it turns out, belongs to a special class of proteins that govern the transfer of genetic information from DNA to mRNA. That means it has the ability to pry open sealed pockets of genetic material, bind to previously silent genes within, and activate them. The discovery provides new insights into how cancer derails a stem cell's carefully tuned decision-making process, thereafter making it impossible for it to make normal tissue. The genome is not an open book. In fact, it is more like a library filled with a few billion books that are mostly under lock and key. The majority of genetic material actually lies silent with non-coding and tightly bound packets of DNA cordoned off by histone proteins in a closed state. As together, the DNA and histones form what is called closed chromatin. The genes that are packaged into this cloistered material is inaccessible to the transcription proteins or factors that would help it to express the genes within. As there are a few rare keys that aren't simply transcription factors, these pioneer factors can unlock those genetic packets. They possess the superpower to appear inside the closed chromatin and recognize binding sites within. They then recruit other transcription factors to help them pry open the closed chromatin and bind to receptor sites on the nucleosome, which reprograms the chromatin and activates new genes. In adult skin, SOX9 is normally associated with maintaining the identity of adult hair follicle stem cells. It is normally suppressed in adult epidermal stem cells, but that is not the case when it comes to basal cell and squamous cell carcinomas. How this process might unfold, step by step, has been unknown. Reprogramming in vitro happens really fast, within 48 hours. With such a short time window, it is hard to get a good resolution on the sequence of events. And to find out, the researchers engineered mice that contained a copy of SOX9 that could be activated in their adult epidermal stem cells when the mice were fed doxycycline, a drug that induced the transgenic SOX9. In adult tissues, choices that were easily made in embryogenesis are tightly suppressed so that adult stem cells stick with their dedicated task. Unleashing SOX9, however, turned out to be a potent influencer, progressively reprogramming the epidermal stem cells to new fates by only expressing the single SOX9 transcription factor. They were able to induce basal cell carcinoma-like structures by week 6. By week 12, they started to see lesions that resembled human basal cell carcinoma. And simultaneously, they tracked the epigenetic process going on behind the scenes. In the first 2 weeks, SOX9 turned off the epidermal stem cell genes, reversing their normal state. They began to turn on hair follicle stem cell genes. And seeking the mechanism, the researchers discovered that to achieve this fate switch, SOX9 hijacked the nuclear machinery from the active epidermal genes and brought this stolen equipment to the silent hair follicle genes. It then enlisted other transcription factors to pry open the closed chromatin bind to the silent genes within, turning them on. When SOX9 could not be regulated, the stem cells failed to make hair, but instead just kept proliferating and activating several new transcription factors, eventually leading to a basal cell carcinoma state. This complicated identity, shifting back and forth, was only possible because SOX9 is a pioneer factor. Only a pioneer factor has the ability to access closed chromatin. And because SOX9 is overly active in many of the deadliest cancers worldwide, the researchers aim to look for ways to intervene in its role in proliferating these cells. And by identifying how SOX9's interacting proteins and its target genes change during malignancy, we hope to make inroads into unearthing new drug targets for these cancers. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.